Thank you very much for allowing us to share our work on COVID-19, the GI tract, and the microbiome. But what do we know today? Here are my disclosures. So today I'm going to cover 4 key areas relating to the role of the gut microbiome on COVID-19 risk and severity, the gut microbiome and development of long COVID, how we could modulate the gut microbiome as potential therapeutics in COVID-19, and what is the impact of the gut microbiome on vaccine response. So although the pandemic has affected over 440 million cases worldwide, leading to 6 million deaths, there are 3 key questions that we have in complete understanding. And that is, why are some people more susceptible to the disease? Why do some have worse outcomes when infected? And currently, how durable is protection from vaccination? It transpires that COVID-19 is not only a respiratory disease. The human gut is clearly a target, as has been shown by several studies whereby virus is detected in various parts of the intestine. And one of the many reasons is that the viral entry receptor ACE2 and the cellular proteases TMPRSS2 is not only expressed in the heart, in the lungs, but also enriched in the enterocytes of the small bowel. And in this regard, many early studies have shown that GI symptoms are not uncommon and can be reported in up to 70% of cases. Interestingly, detection of the virus in the stool does not always correlate with GI symptoms, hence there may be some asymptomatic carriers. Stool analysis has also confirmed the presence of the genomic and subgenomic RNA of the virus in affected patients, more so in those with severe disease. In this study, they looked at over 200 patients in multiple clinical specimens and found that the most likely tested positive are the lower respiratory tract samples. However, live SARS-CoV-2 virus was also observed in the stool sample from 2 patients who did not have diarrhea. Although the isolation of live virus has rarely been successful and that GI infection is mostly self-limiting, there has been a new concept which we learned from HIV infection, whereby the gut may act as a viral reservoir, in that in the gut there's SARS-CoV-2 virus infected immune cells that may circulate to the bloodstream via immune cell trafficking, leading to recurrent disease or continuous immune stimulation. And several observations have shown persistent infection of intestinal epithelial cells for prolonged memory immune responses and the future prevention of reinfection, and this clearly is an exciting area to explore further. But at the same time, the human gut is also home to trillions of microorganisms and considered one of the largest immunological organs in our body. And apart from regulating immunity against bacterial infections, we have data showing that it also defends against viral infections such as influenza by the gut-lung axis, and this is clearly applicable to COVID-19 infection. In the early pandemic, our group has the opportunity to look at alterations in the gut microbiome of patients who were hospitalized with COVID-19. We collected fecal samples every 2 to 3 days from those who were admitted, and we were able to expand this cohort to over 100 cases with mild, moderate, severe to critical disease severity. And we had a control group of non-infected patients and also a positive control group of pneumonia patients. We captured their standard diet as well as perform microbiome profiling using deep shotgun metagenomic sequencing. One of the first observations we found was that amongst all the host factors known to affect the gut microbiome, COVID-19 had the biggest magnitude, followed by hyperlipidemia, pneumonia, antibiotic use, and other comorbidities. There are several observational studies who consistently show that in patients with COVID-19, there is decreased bacterial diversity, increase in opportunistic infections in the gut, and a diminishing symbiomes. When we look further at those who had none or low SARS-CoV activity in their gut, we found that they harbor more beneficial bacteria to the host that can produce short-chain fatty acids. In contrast, those with high SARS-CoV infectivity are the ones who had more opportunistic infections. Some of these pathogens were derived from the oral cavity. There were also 4 bacteroides species that showed significant inverse correlation with the fecal SARS-CoV-2 load, and all these species have been reported to downregulate ACE2 expression in the murine model. Now we know that pre-existing disease such as diabetes, obesity, heart disease, hypertension, and comorbidities are risk factors for worse COVID-19 outcomes. But interestingly, these are the people who also have been found to have depleted certain bacteria such as Bacteroidetes and the short-chain fatty acid producers. So suggesting perhaps depletion of Bacteroides species may increase host susceptibility to SARS-CoV-2 infection. One another question is if indeed that the virus is in the gut, are there any evidence of antigen persistence or viral replication? We hypothesize that if indeed SARS-CoV-2 did infect the gut, then we would be able to identify certain viral RNA activity. And by using metagenomic sequencing, we found that high 3N coverage was seen in the gut of all these patients who had COVID-19, suggestive of active activity. And in vitro transcription analysis of SARS-CoV-2 infected cell models have shown that this 3N of the SARS-CoV-2 genome was highly covered, which indicates active viral replication and infection. What was interesting is that when these patients have cleared their SARS-CoV-2 virus in the nasopharynx, we could find high activity with these spikes detected days and weeks after they have recovered, again suggestive perhaps prolonged activity of the virus still in the gut. The long-term implications of this is not clear as yet. Now, which is chicken and which is the egg? How does alterations of the gut microbiome affect the COVID-19, or is it vice versa? Harris Soker and groups has looked at non-human primates and challenged it with the SARS-CoV-2 virus, and they found that from day 0 up to day 13, there were significant alterations of the composition as well as the functions of the gut microbiome. And proteobacteria and firmicutes were positively correlated with the virus in the respiratory tract. Following that, a group has looked at nosebiotic mice and colonized them with different bacteria. And what they found was intestinal ACE2 expression levels were significantly higher in the germ-free mice compared to conventional mice. And the microbiome was also associated with reduced GI and respiratory tract ACE2 expression. Now, when the microbiome was depleted using antibiotics, it partially recapitulated the germ-free phenotype, suggesting that microbiome-mediated regulation of ACE2 expression may be an important factor to consider. To take this further, we started studying the gut microbiota composition in patients with COVID-19 and how these reflect disease severity and their immune response. In over 200 cases, we were able to identify several gut commensals which is known to have immunomodulatory functions that were underrepresented in COVID-19 patients. And you could see here in those who had more severe or critical disease, these beneficial bacteria were clearly completely depleted. And also those with higher relative abundance of these species actually had a lower concentrations of inflammatory cytokines in their blood. Hence, there were several bacteria that were associated with increased inflammatory marker concentrations. Overall, supporting the notion that gut microorganisms may influence the inflammatory status of the host in COVID-19 infection. When we followed these patients for over 30 days, it's rather alarming to see that in those who have recovered, their gut microbiome profile remained distinct from those who had no COVID infection. And when we further interrogate the impact of antibiotics, as some of these patients may have received antibiotics, you could see here that positive patients who had received antibiotics, their gut microbiome profile had a further shift from the healthy state, and they had lower levels of Eubacterium rectali, Rosebaria, as well as the Bifidobacteria colonizers compared with the non-COVID controls, suggesting antibiotics perhaps should not be used more liberally in some of these patients. Zandfeng, one of our postdocs, were interested in the metabolites of these patients, and she used targeted and non-targeted metabolites to understand whether short-chain fatty acids biosynthesis were affected. And indeed, she found that in these COVID patients who had lack of short-chain fatty acids and also the L-isoleucine biosynthesis, it correlated with more severe disease and also increase in some of the plasma cytokine levels. And since then, there have been some mechanistic work looking at the supplementation of butyrate to protect the host from viral infection via the downregulation of the genes essential for SARS-CoV-2, such as ACE2. Apart from the gut bacteria, the microbiome also consists of other dark matter, such as the virus as well as the fungi. So one of the scientists used metagenomic sequencing to understand the human gut RNA and DNA virome. And what was identified is that the fecal virome in SARS-CoV-2 infection actually harbored more stress, inflammation, and virulence associated gene encoding capabilities, as shown here in red, as well as virulence associated with their bacterial hosts, in contrast to those non-COVID-19 patients. And whether this may translate to further risk of increased bacterial and viral infections warrants further study. We also found that the fungi communities of those hospitalized patients were substantially altered. In other words, they had enriched of fungal pathogens, especially from the general of the Candida, as well as the Aspergillus, both in severe as well as non-severe cases. And the unstable gut microbiomes and this prolonged dysbiosis also persisted in a subgroup of patients who have completely recovered from the infection. To take this to a different level, Qing, one of our scientists, started looking at network analysis of the three categories of bacteria, fungi, and virus. And this network matrix were able to depict the influential impact of the microbes on the integrity. There were the top 10 bacteria, virus, and top fungi that actually clustered into two separate groups. And the more severe clusters had more pathogenic microbes, including the Candida, Klebsiella, and Crocerium family, and the integrity was also very much impaired. So understanding now the alterations in the gut microbiome, do they also play a role in post-acute COVID syndrome? Following the acute infection, subgroup has now established that a substantial number of patients continue to have persistent symptoms known as long COVID or post-acute COVID symptoms, ranging from fatigue to cough to anxiety, depression, as well as sleep disturbances. The exact cause or mechanisms of this is not completely known. We hypothesize that the gut microbiome may play a role, and we followed patients from admission until 18 months after they have recovered. And in this cohort of over 200 cases, about 76% developed post-acute COVID syndrome, and the top most common symptoms were fatigue, poor memory, and hair loss in this cohort in Hong Kong. We found that patients with long COVID had distinct gut dysbiosis that persisted for over 6 months. And you can see here the yellow are those with post-acute COVID syndrome followed to 6 months, and those with no post-acute COVID syndrome were rather similarly clustered with the non-COVID controls. And the gut microbiome of patients with post-acute COVID syndrome were also characterized by higher levels of Ruminococcus, Bacteroides, and also lower levels of Faecalibacterium prausnitzii. What was surprising is that the gut microbiome pattern could in fact reflect different symptoms in patients with long COVID, as listed up here. In those who had persistent respiratory symptoms, there were an increase in opportunistic pathogen related to pulmonary diseases, and neuropsychiatric symptoms were characterized by nosocomial gut pathogens, such as the Crocerium families. There may also be a prognostic role of the gut microbiota in predicting risks of long COVID. When we analyze the gut microbiome at admission of these cases, you could see here that those who eventually developed long COVID, PATS, or those who did not, were very distinct in their clustering. And there were some top 10 bacterial species incorporating into clinical characteristics and different matrix that may provide a predictive tool for some of these patients. And with this work, it's now the media has disseminated and highlighted the importance of the intestinal bacteria in COVID-19 severity. And also, perhaps we may consider how manipulation of the gut microbiome may help to reduce the long-term complications of COVID-19, as highlighted by new scientists and other groups. So could we modulate the gut microbiome as a potential therapeutic in COVID-19? There are many ways in doing so, ranging from simple measures of dietary therapy to probiotics, prebiotics, as well as different drugs. Here's an elegant study that provides indirect support for the potential role of dietary intervention in COVID-19. In a multi-center study of over 7,000 subjects, those who had well-controlled blood glucose had higher and better outcomes with COVID-19. In contrast, those who had poor glycemic control had higher mortality, highlighting the feasibility of a low glycemic index either with dietary interventions in order to improve outcomes in hospitalized patients. At the same time, Chang Ling, our assistant professor, developed a new microbiome immunity formula using machine learning and big data and tested it in the pilot study in Hong Kong, whereby half of the patients received the SIM01 microbiome formula and half received just standard therapy. She followed these patients for up to 4 weeks and then measured their gut microbiome as well as assessed their clinical evolution. The first observation was that those who received SIM01 in this pilot study more developed neutralizing antibody compared with the standard therapy. And when she analyzed the blood parameters of these patients, those who received SIM01 also had significant reduction in IL-6 as well as TNF-alpha and several of the other blood chemokines and cytokines. And right now, there are ongoing clinical studies of the gut microbiome modulation in COVID-19 in clinical trial registry. As can be seen up to now, there are 14 interventional trials using probiotics, symbiotics in different dosage, different duration, targeting either acute COVID-19 or for prevention of long COVID symptoms. And these studies range from 40 to over 400 in terms of sample size. And we further await some of these data in due course. There are 2 cases that have been treated using fecal microbiota transplantation, which was published in Gut. These 2 cases are patients with recurrent Clostridial infection, but also had SARS-CoV-2 infection. And following FMT, reassuringly, their fever subsided and their pro-inflammatory markers reduced very rapidly. And in order to identify the role of FMT in COVID-19, we clearly need larger studies to confirm its safety and its efficacy. But there's also a caveat that during this pandemic, we know that the virus is present in asymptomatic people. Hence, screening for FMT donors becomes a real concern in that we need to ensure that stool testing is done in every single donated material before performing FMT. So what about the impact of the gut microbiome on vaccine response? The vaccines are very effective in reducing severity, but we are now seeing breakthrough infections in those who are vaccinated, including following the 3rd dose. Consequently, the Swiss cheese model still holds in that there's really no single intervention that is perfect in preventing the spread of the virus. And if you look at our antibody titer as a sink full of water, you could see that over time, your antibody titer will drop and the risk of COVID-19 will increase. And following the booster jab, the antibody increase and the risk reduce. This risk is never going to be 0, but we want to keep it as low as possible with vaccination. The Clarity study in the UK has shown that with every 10-fold increase in antibody titer, there is an 0.8-fold decrease in the risk of COVID-19. In this systematic review published in the Lancet just this month, they look at the duration and effectiveness of vaccine against SARS-CoV-2 virus, and you could see across different vaccines from AstraZeneca, Moderna, the Pfizer-BioNTech, clearly in all ages and any infection, the waning of antibody and vaccine efficacy really does sort of reduce over time up to 6 months. Apart from the vaccine dosage and repeat vaccine and the route and antigen dose, there are other factors that also influence vaccine immunogenicity, such as the gut microbiome. And here are some of the potential mechanisms by which our gut microbiome could modulate vaccine immunity, from innate sensing of the microbiota by the receptors, enhance antigen presentation by dendritic cells, immunomodulatory microbiota-derived metabolites, or even B and T cells cross-reactive epitopes that are encoded by the microbiota. And there are animal studies and also human studies to show that composition and functions of the gut microbiota are clearly crucial in modulating the immune response of vaccination to flu vaccine, to BCG vaccine, and in a study, the antibiotics-driven gut microbiome impairment can actually alter immunity to vaccines in humans. So we had the opportunity to understand how our gut microbiota composition is associated with SARS-CoV-2 vaccine. And in Hong Kong, when patients received the inactivated or the mRNA vaccine, we followed these patients following their second dose and analyzed their stool microbiome as well as their neutralizing antibody. And we followed up to 200 cases now. We found that the baseline gut microbiome and functional biomarkers were associated with response to vaccine. For instance, the early colonizers of our bacterium, which is Bifidobacterium adelanthitis, when they are present in high abundance in the gut, in vaccinees who received the inactivated coronal vaccine, they actually had a higher antibody response. And for the inactivated, for the mRNA vaccine, we found that higher relative abundance of bacteria with flagellum in the baseline gut microbiome was associated with, again, higher antibody response. And Rosebery feces is one of the major contributors to gut bacterial motility as well as febrile flagellum, highlighting there may be a role of adjuvant therapy with vaccination using bacteria or microbiome modification in these patients to improve immunogenicity and reduce antibody waning in the longer term. There are some patients who develop side effects following COVID-19 vaccine, although these are temporary. And we found that Prufitella copri, one of the fiber-degrading community, were enriched in individuals who had fewer vaccine adverse effects. And these bacterial species may play an anti-inflammatory role in host immune response. There are now ongoing observation studies of gut microbiome and COVID-19 vaccine. For example, in the United States, there's a large study of 10,000 subjects looking at the impact of the gut microbiome and function on efficacy of COVID-19 vaccine. So in summary, what do we know today? Our group and others have shown gut dysbiosis is concordant with disease severity in COVID-19, and persistent dysbiosis is evident following viral clearance, and we need to understand the long-term implications of this. An individual's gut microbiome profile may affect susceptibility to long-term complications of COVID-19, and the composition may affect and predict immune response to vaccine and vaccine-associated adverse effects. But just like a double-edged sword, there's several things we don't know, and that is the impact of COVID-19 on our neonatal microbiome. Will this pandemic cause infants their microbes? With social distancing, with the hygiene status being so vigilant, are we wiping out the good guys inside our gut microbiome? So how do we balance microbiota gain versus microbiota loss in response to this COVID-19 pandemic? If the hygiene hypothesis holds true, would we be seeing more increase in immune-mediated disease in the next 2 decades or so? And we also need to understand how new variants, such as Omicron, affect the gut microbiome. Nonetheless, there's a silver lining in that emerging studies are showing a mechanistic link between SARS-CoV-2 infection and the gut microbiome. And we may be able to leverage on these in utilizing microbiome-based profiling as a potential tool in early risk stratification and severity of long-term complications. And microbiota targeted intervention in the acute setting and to optimize COVID-19 vaccine antiviral response may become possible. To move this forward, we need to move from some of the association studies to mechanisms and in the future clinical application. And I'm very proud and grateful to every single member of my team and my collaborators who have contributed and dedicated their time effortlessly to this work that you are seeing here. And we hope we can further advance the knowledge in this field in the years to come. And thank you for listening, and I hope to meet you in person one day.